Okay, people, let's talk about pure culture. The best way to get into this is to think about the opposite. Let's think about a mixed culture. And the thing about mixed culture is you can find them anywhere. So if we draw our table, so this is a tabletop, and we're down at eye level, just looking across the surface of the table. What you would see is you'd see some purple bacteria. If you had amazing eyeballs that could magnify bacteria, you'd see some purple ones and some blue ones, and the color isn't important. Bacteria are too small to have color. That color is a physical impossibility for small objects like that. But what I'm trying to show you is that they're different. And they are everywhere. Every surface you could imagine, as long as it's like a normal temperature and, again, not in a laboratory, any surface has bacteria on it, but lots of different bacteria. So why do we care about this? Why do we care? Well, we want to do research. We want to do things like figure out what microorganism is causing your patient's disease. Well, how do we do that? If you take a sample from a person, from their skin or from their mouth, you get a mixed culture. You get a huge different number of bacteria. What we need often is to do something like what we did with Koch's postulates, where we have to isolate the one bacterium we care about in pure culture so that we can study it. In the case of Koch's postulates, once we have that one bacterium in pure culture by itself, if we inject it into a sheep and the sheep gets sick, we know that one bacterium is the one that causes the disease. If instead we have a group of like 10 different types of bacteria mixed together and we inject them into a sheep and the sheep gets sick, we don't know which one caused the infection. So there are just a lot of reasons why we would want pure culture. So in the laboratory, what we will often have in the laboratory is test tubes with growth medium in them. And if we just leave a test tube with growth medium, so if this is nutritious broth that bacteria can grow in, if we just leave this open, like dust can fall into it, and if dust falls into it, there are a lot of different microorganisms in dust, and they will fall in. So what you get is a purple cell in there, and again, the color, it doesn't matter, it's just some bacterial cell, and it's going to be happy. It can use the carbon and nitrogen that's dissolved in the water in this broth as nutrition, and it's going to grow. It's going to double again and again and again, and make a lot of purple cells. But at the same time, the blue ones fell in, and they're going to do the same thing. These are like cocci, and they're going to keep growing and growing and growing, and they all get mixed together. And then some of them are like blue bacilli. They're going to do the same thing. And so what you get ultimately is a mixture, but you can't tell by looking at it that it's a mixture. All you see is a cloudy tube. This would look like a cloudy tube, and you can't with your eye see any of these individual cells. They are far too small for your eyes to see them. And you're not going to put a microscope up against the side of this tube when it's realistically got billions of cells in it. So the real number of cells in a cloudy tube is astronomically huge. So this would be a mixed culture. So that's a mixed culture. But it's also possible in the laboratory, under the right conditions, if we are careful, and if we have a cap over our tube so the dust can't fall in, if we introduce one cell, and that's it, if we just put that one cell in there, well, yeah, it's going to double and quadruple, and it's going to keep doubling until these cells run out of food. But all of the cells in this tube are descendants of that one cell, and they are clones of each other. Minus a few random mutations, they are all clones. And that's the closest thing we can get to pure culture. So it is possible to have a pure culture in broth. The question is, how do we go from there to there? And that's what we're going to see in a later video. We're going to see how we get from there to there. But before we do that, we need to go back and look at our petri dishes again. We need to think about agar again, because agar is going to give us the tool we will use for getting a pure culture. So again, I'll draw a petri dish that has solidified nutrient broth in it. So let's imagine you put one cell on this plate and then you put the lid on. Well, that one cell will reproduce until there's a giant pile of that one cell and its clones, and that's a colony. That's a pure culture colony right there. That's a beautiful thing. That's what researchers want. What is also possible is the opposite of that, which would be you could have an actual mixed culture in the same colony, and that sucks, but that is a real thing that can happen. So what if, instead of putting down one purple cell, I put down a gob of cells? So I had some sample I took from the environment that was visible, some clump, like, I don't know, a piece of fecal material or something that is just full of bacteria. Well, there are going to be a whole bunch of purple cells, and different ones, all mixed together, and yeah, they're going to all replicate until you have one colony, and you might or might not be able to tell by looking at it that it's a mixed colony. The problem is we can't always tell, because most cells, I mean, the bacteria just don't have any color, and so what you get is some kind of colony that looks just like a pure culture colony. So we need special techniques for making this so that we know we have pure culture, because once we have this, we can take those cells. Whoa, what just happened? Once we have this, we can use certain tools to make this. We can take these cells that are clones of each other and put them in a tube, and they'll grow up and they'll still all be clones of each other, and all their descendants will be clones. And that's what we're trying to do in a laboratory. So one of the most fundamental techniques used in the laboratory generates pure culture using agar. And from there, we can do lots of other things. So we will get there shortly.